Good evening, everybody. My name is Mohanraj Krishnan, and today I'm going to give you a brief presentation of my PhD project entitled A Model of Childhood Obesity. Obesity is a major public health issue and is associated with long-term increased risk of heart disease, diabetes, and hypertension, all of which are consistent risk factors of the metabolic syndrome. These days, children are becoming more obese, and they are also presenting with similar symptoms early in childhood. And it's no different in New Zealand. In 2006, the New Zealand Health Survey showed that the obesity rate in children was 8% and has risen to 10% in 2012. Furthermore, there are ethnic differences, with Maori and Pacific Island children showing higher rates of obesity compared to Caucasian children. Now, there are a number of factors involved in obesity, including intrauterine factors, genetic factors, environmental factors, and microbiology environment of the gut. And it's likely all of these factors play a role with each other to contribute to the onset of obesity. However, my project will focus on polymorphisms, which are single unit changes in the DNA code, which might slightly alter the gene function. Furthermore, I'll be looking at diet and exercise as possible intervention mechanisms to reduce childhood obesity, and whether these environmental factors may modify the genetic risk of obesity. I would like to introduce to you the Thrifty gene hypothesis, proposed by James Neal to explain the modern emergence of obesity. Genes that promoted obesity were considered advantageous in early human history, where starvation and migration were an issue. However, in today's modern society, where there is constant availability of food and decreased physical activity levels, these genes that were once beneficial for our survival are now detrimental. This results in disruption of gene-environment interactions, resulting in the development of modern chronic diseases, including obesity. This diagram here shows the Thrifty gene hypothesis during ancient times. Early humans went through cycles of feast and famine, and during feasts, these individuals would store fat in preparation for the famine phase. Humans that had the Thrifty genes were more efficient in storing fat and utilizing fat, and were more likely to survive during famine. Likewise, these humans also went through physical activity cycles, and these individuals that also had the Thrifty genes were more physically capable of surviving the hunt and gather, and were also more likely to survive the famine phase. Combined, both these processes provide a greater drive for Thrifty genes. These genes were passed down to their offspring, and subsequent generations had a greater chance of surviving the next famine phase cycle. However, in modern society, there is no famine or hunt or gather. Instead, it's replaced by constant food availability, and with industrialization, reduced physical activity levels. This creates a mismatch between the genes that have evolved to protect us from starvation with today's surplus environment. This results in disruption of cycling, and results in excessive Thrifty storage, which gives rise to obesity. If we can intervene in this target area here, we have the potential to restore proper cycling, and thereby have a greater chance of reducing obesity. Therefore, my project is investigating associations between polymorphisms with obesity in the children of screening for pregnancy endpoints cohort, where these children are separated into three main categories, into normal, overweight, and obese. Furthermore, I'll also be looking at whether physical activity and diet can be used as possible intervention mechanisms to reduce childhood obesity, and whether these environmental factors can actually compensate for the effect of obesity-related genes. My study is a cross-sectional survey of 6-year-old children with detailed pregnancy data collected at 15 and 20 weeks, late pregnancy, and after delivery. Currently, I've extracted DNA from either saliva or blood from these children, and will be using sequenom mass array to measure this. And sequenom mass array is an experimental tool designed to detect differences in polymorphism. This is a scatter plot output file from a sequenome mass array. Each dot here represents an individual, and each cluster represents a different polymorphic combination. We will compare the difference in frequency between each cluster between our obese cases and normal controls and see whether our selected polymorphism is associated with obesity. Currently, I've also been identifying genes involved in obesity and separated them into three main categories. Adipicity storage, and these genes are involved in fat accumulation and distribution. Appetite regulation, and these genes are involved in feeling of fullness, energy expenditure and behavior, and fetal growth and birth weight. And this last category is potentially interesting, as studies have showed that children, babies born bigger at birth, have a greater risk of becoming obese later on in childhood. I want to highlight this diagram here to show that a polymorphism may have most polymorphisms have a modest effect, and that they could either protect from or confer susceptibility to obesity. However, it's a combination between genetics and environmental factors that drive most of the population to an obese phenotype. Ideally, if we can shift this distribution to a normal state, we can reduce obesity, and one way we could do this is to modify lifestyle factors that we can control for, which includes diet and physical activity. Therefore, we have collected 1 week's worth of physical activity data from these children using accelerometers, which measure acceleration peaks. This is an actigraph, which is an output file of these accelerometers, and each peak here represents an acceleration over 1 minute time interval over 1 day. We have separated each of these into three main categories: sedentary, moderate, and vigorous. And we want to compare if level or intensity of exercise directly correlates to levels in fat mass. We also want to see if any form of exercise can actually compensate for a polymorphism outcome or polymorphism effect. We've also collected dietary information from a 24-hour diary, food diary, a food frequency questionnaire, and family questionnaire. And these surveys collected information regarding general dietary habits, nutritional awareness, frequency of meals, and the specific types of food they have in their household. And we'll be also using this dietary information to estimate how diet may modify the genetic risk of obesity in these children. Hopefully, if proper routine exercise and a balanced diet can restore proper cycling and normalize Thrifty gene storage, this has a chance, possible potential mechanism to reduce childhood obesity. So my study offers a unique opportunity to advance our understanding between environmental factors and genetics in the context of childhood obesity. And although there is a fixed genetic risk, we know that early intervention in life by modifying our lifestyle factors has a better success rate if we intervene later on in life. This study also provides the tools to understand how genes may respond to environmental factors, which can result in differences in body mass, in fat levels, and also obese and lean phenotype. I would like to thank my professors, Associate Professor Andrew Schelling, Leslie McCowan, and Dr. John Thompson, also Dr. Lindsay Cree, and everyone who participated in the Children of Scope study, including the parents, the children, and the recruitment nurses, and all other researchers, and the members of the Schelling, Cree, Chamley, and Print Lab. Also like to thank my funding body, HRC, who's funded this project, and Gravita, who's funded me to undertake this project. Thank you very much.